All right, welcome everyone to round three of the SJSU College of Science virtual seminar. Um, we're hopefully creating a tradition here, which is really exciting. By the way, if you're on the call and you're interested in giving a talk at one of our future Friday seminars, please let me know. I'm Michael Kaufman. I'm the Dean of the College of Science at San Jose State. I see we have plenty of people currently from San Jose State, including faculty and students. I see at least one retired faculty member who's reached in, and I see a lot of names that I assume are friends and colleagues of our speaker, and very, very excited to have you all here today. Today's speaker is Dr. Phil Heller, who, let me tell you a little bit of his bio. Phil is a native San Franciscan who received his Bachelor of Science degree in electrical engineering way back in the pre-space shuttle days, I think is the way I frame it, and joined the Silicon Valley workforce at a time when some really interesting things were going on. First as a programmer, then as an author and speaker. He was involved in several startups, including Sun Microsystems and Next. I think this is my favorite thing in his whole bio. He has personally been fired by Steve Jobs. So that's a badge of honor right there. In 2006, he heard about bioinformatics and decided it was time for a career change. So he came to San Jose State to get a master's in computer science, and then moved on to UC Santa Cruz, where he got his PhD in bioinformatics with an emphasis on marine ecology. He graduated from Santa Cruz in 2014, spent a postdoctoral year at Moss Landing Marine Labs, where many of you are currently, and then came back to San Jose State several years ago as an assistant professor in the computer science department. In addition to his academic and industry work, he is also the author of 5 books about programming, and according to his bio, also one unpublished novel about a dragon. And I don't think we're going to hear about the dragon here today, but I'm really thrilled that Phil is able to present to us today to talk about his work that's at the intersection of computer science and marine science, with the title Software Without Algorithms, Deep Learning in the Deep Ocean. So without further ado, Phil Heller. Oh,
Thank
and
you.
I'm sorry, I apologize, Phil. Let me just repeat the instructions to people. There is a Q&A button at the bottom of all of your screens. If you have a question for Phil, please type your question in the Q&A. If it's appropriate for me to interrupt Phil and have him answer the question on the fly, I'll do that. Otherwise, we'll hold the questions for the end of the session, and I'll let people who ask questions ask their own questions in their own voices. So thanks a lot, and let's get started.
Thank you, Dean Kaufman. Michael, not only for that introduction, but also for arranging for me to speak today, and in fact, for arranging this entire seminar series. I think we're all a bit frayed by the challenges of these times, and I know I am, but there's a lot of support to be felt in events like this when we come together as a community of scientists and people interested in science to learn about the world and to come to evidence-based conclusions about it. The warmth and welcome that you add to these events has an effect beyond the knowledge that gets shared. In that spirit, I'd like to dedicate today's talk to all teachers and administrators at all levels who are struggling to keep the light of education shining through this year's darkness. Today, I know the blurb said 3 projects. I'm actually going to tell you about 2 projects in my research group, both based on deep learning. Now, no doubt you've heard the term deep learning, though you might not know much about it, but it knows about you. Last year, I visited a friend, and while we were preparing dinner together, he had his online music provider play a channel based on our common musical tastes. You've probably seen these for a while. You train the channel by telling it what you like and what you don't like, and after that, the channel has you figured out and chooses the songs autonomously. While we were cooking and listening, his 11-year-old daughter played a game on her laptop in a browser crammed with ads targeted to her demographic. That means a lot of pink, a lot of purple. And then over dinner, my friend's wife told me that she was just home from Beijing, where she has a bank account. She'd gone to the bank to withdraw some funds. At no point during the transaction was she asked to show any identification. Cameras had observed her and software had analyzed her from the moment she walked into the building. By the time she reached the front of the line, her identity was established. All of these situations, the music service, the browser ads, and the bank, are driven by deep learning software. And whether you find these situations convenient or irritating or sinister, deep learning is a part of our everyday world, but it isn't well understood outside the computer science community. So today I'm going to do something unusual for this kind of talk. I'm going to spend a while on a lay explanation of deep learning. It's a good thing to understand because it's embedded in our society. And also, for those of you who are wet lab scientists in exile from your wet labs, deep learning and other software analysis techniques may be a way forward for you until your labs reopen. Computer science collaborators may be able to help you analyze data that you've already collected, but maybe these techniques can help you look at your data from a new direction. And after we have an understanding of deep learning, I'll tell you about my new projects, which use deep learning to study various aspects of marine ecology. Deep learning is an extension of neural networks, and that's a concept that originated in the 1940s. The big idea of neural networks is to model thinking. And the idea is that make a thinking machine analogous to a brain. How do we do that? Well, maybe our brains do execute algorithms, but it doesn't feel that way. Humanity's collective intuition about how our brains work, about how we learn stuff after tens of thousands of years of doing it, is that we somehow learn stuff and later we somehow access what we've learned. That's all. The only algorithm-like things in our brains are neurons, nerve cells that are massively connected to other nerve cells. Neural network technology, including deep learning, starts by representing a nerve cell in software. That's the algorithm, and then scaling up. So, okay, this technology isn't completely algorithm-free, but the massively replicated neuron algorithm is so trivial that it barely justifies the name. It's really just arithmetic. So here's a schematic of an artificial neuron. At the heart of it, in the big bubble, we have some math and some logic, and it has some inputs. And I've tried to draw this so that you can see that these inputs might represent simulated sense organs from the 5 senses. And because this is computer science, we love reducing everything in the world to ones and zeros. So all the inputs are values that are either zeros or ones, and the outputs are either zeros or ones. Now, this is a primitive sort of artificial neuron. More modern ones are more sophisticated, but we don't need that sophistication in order to get you the big idea. Well, is this realistic? Imagine a primitive metazoan, that's an animal, with an early sort of proto-eye that has 5 inputs coming into the neuron, 5 senses, and let's decide that the inputs are 0, that is, they're not stimulated, if there's nothing out of the ordinary in front of this animal. And they're 1 if these inputs are stimulated. And the neuron itself has this algorithm, if that's the word, that just says, if 4 or more of my inputs are on, I'm going to turn on my output. And maybe what the output does is trigger a function in the brain that gives this organism a little squirt of adrenaline. Because if what's occluding the inputs is a threat, is a predator, then a little bit of adrenaline to get out of the way is going to be a very useful thing. Whatever DNA makes this trait happen will be supported by evolution and will tend to survive. And in fact, the mechanism that does this will tend to become more sophisticated over time and possibly even turn into our modern eyes. Now, neurons actually don't exist individually. We connect them all together. This is what we would call a neural network, although for this century, this is actually quite small. This is just to give you the idea that we can take all the outputs from our first layer of neurons over on the left, feed them as inputs into another neuron, and keep that going. Looking closer at a single neuron, if we say input 0 equals biologically off, input 1 equals biologically on, and as I say, that's the simplest implementation. And the output is 0 or 1, and that's just a very simple arithmetic function of certain learned parameters called biases and weights. And for now, let's just say learned is going to go in these finger quotes, and we'll get to them later. There's one weight associated with each input. The algorithm says for all the inputs that are on, we'll add up the weights. And if the sum is less than the bias, I got these backwards. If the sum is greater than the bias, then the neuron fires. Otherwise, the output is 0, the neuron doesn't fire. So that's pretty simple, right? You choose a few inputs, you add up, if the ones that are on, you add up their weights, you compare them to a number, and you fire or not. This is why I say it's barely an algorithm. But what are these parameters? What are the 5 weights that I've drawn in blue? What's the bias that I've drawn in purple? Well, figuring these out is the secret sauce of neural networks. And in fact, if you've heard the related expression machine learning, anytime we have a piece of software or a statistical model that tries to imitate some part of the physical universe and does it by having a whole bunch of parameters that need to be figured out, learning these parameters is what makes machine learning. And of course, that's the secret sauce. That's the hard part. Well, neural networks actually date back to even longer than I do. And they have an interesting history. Right around the 40s and 50s, the thought was, well, brains are made of neurons and brains think. So we'd love to make thinking machines, because in those decades, they weren't really computers as we know them. So the thought was, if we can make mechanical neurons, maybe we can hook them all together and make something that thinks. And here's the output of that thought process. This is the Mark I perceptron from 1958. Filled the whole room, cost just over $2.5 million in 58. That would be 22 million today. It couldn't do very much. I want to show you a picture of my hero, Frank Rosenblatt, its inventor. And it's a little hard to see, but at the tip of this arrow here, he's debugging, or you might say he's doing brain surgery, with a wire cutter. Yeah, those were the days. Well, in the 1960s, computers had invented, and it occurred to people that maybe we could implement these in software rather than building them out of wires. And interesting results were gotten even with a single simulated neuron. And there was theory about what we could do if we had more than one of these, but computers just weren't powerful enough to actually implement the theory. Then in the 1980s, this technology had moderate commercial success, especially with recognizing handwriting. And there was a boom and bust. It's called the AI winter, AI being artificial intelligence, because really no company made it big based on this technology. It just wasn't quite time. But then in the 21st century, people, especially at Google at first, started thinking, you know what? We really improved computers in the past 20, 30 years. Let's try again. Computers were big and fast enough now, and there was success now in some application domains, especially, this is really important, in image recognition. And if you've heard of TensorFlow, this was the first publicly released software library to do deep learning. It's the first, it's very powerful. I find it a bit difficult to use, but I think everybody knows this name. Speaking of handwriting analysis, let me just show you an example of how a deep learning neural network can recognize handwritten digits. What we see here is the neurons, those are the bubbles, arranged in 3 columns, which we're going to call layers. We've got 15 neurons, 15 in what we call the hidden layer, that's the one in the middle. The output layer, we have 10, that's not a coincidence. And on the input layer, I've drawn 8, but there's actually 784 of them. And if you're wondering why such an odd number, 784 is 28 times 28. And 28 by 28 square grids are the conventional way to capture individual handwritten digits when people are filling out forms. And the way these digits are entered into any analysis system is these 28 by 28 pixels, so 784 of them, are converted, in the simplest case, to a representation that's 0 or 1, depending on if the little square is black or white. And these 784 ones and zeros become the inputs to the first layer of neurons. So back to our diagram, the change over on the left is intended to mean that 784 ones and zeros are going into the 784 inputs on the left. What we want is on the right. You see how all the outputs are 0, except for one of them, which is a 1. So imagine the top output represents, I just saw a 0. Next output represents, I just saw a 1. This last one is on because hopefully this network will say, I just saw a 9. And none of the other outputs will light up, will turn on, will become a 1. This is a clean classification of the input is a 9. Okay, all we need now, we've almost solved the problem. There are 809 neurons. Every one of them needs a bias. And the input, every input of every neuron needs a weight. So for the hidden neurons, there are 15 hidden neurons. They've each got 784 inputs, so we need 11,000 something numbers. And then we also need a weight for each of the 15 inputs to each of the 10 outputs. We need another 110 numbers. We need to compute optimal, or at least optimal enough values for each of very many numbers. And if we can do that, then we have a classifier that does what we want. If we can't do that, we have a classifier that doesn't do what we want. So as I've said, the secret sauce comes from figuring out the values of these biases and weights. How do we do this? I don't want to get too deeply into the technology of how we compute biases and weights, so I want to approach it by analogy. This is from Discover magazine in 2011, and it's an article about training dogs to smell the byproducts of lung cancer in somebody's breath. Now, this kind of makes sense to me because if there's a tumor in the lung, then it makes sense that its byproducts, some of them would be exhaled. And dogs have amazing noses, so it makes sense. And the way you train a dog is exactly how you would figure it out. You have the dog sniff many training samples, and eventually it's good at detecting that odor elsewhere. And of course, whenever it does detect that odor, you reward it. And if it doesn't detect the odor, if it says, no, that's not cancer when it is, or if it says that is cancer when it isn't, when it's a tennis ball, then you say, no, you got that one wrong, let's try again. And this was how I planned to make this point until 2 nights ago when I was reading the Washington Post and I saw this. So it turns out dogs are being trained to sniff out coronavirus in pretty much the same way. So how do you train a neural network to recognize handwriting? Lots of different algorithms, lots of different approaches for doing this. They all begin by initializing the various parameters, all those biases and all those weights, to random numbers. And then you expose the inputs to a whole lot of zeros, and a whole lot of ones, and a whole lot of twos, and so on. This is like telling the network, these are zeros, these are ones, these are twos, or like telling the dog, this is cancer byproduct. And every time the network gets it wrong, you correct it. You adjust the parameters based on response to the training examples. And if your adjustment improved your network, then you keep on going and keep adjusting in that way until your adjustments aren't helpful anymore. And then you find another way to try to adjust it. So that's how we train deep networks. And now I want to talk about a couple applications to marine ecology. The first project, which is well underway, is called peripheral vision. And the second one, which is massive and has made great progress but has a long way to go, is called coral vision. So let's start with peripheral vision. If this image looks familiar to those of you who were here last week, there's a lot of overlap with this project with the presentation that we heard from Dr. Amanda Kahn from Moss Landing last week. And in fact, she and I are collaborating on peripheral vision. If you've forgotten, or if you weren't here last week, peripheral vision is about glass sponges. They're the phylum Porifera, so I hope you'll forgive my pun, the class Hexactinellida. These are ecologically important, and especially so during climate change. The ecological importance, I don't want to spend too much time repeating what Amanda said much better than I could last week, but I want to point out that like corals, which you can already tell I also care about, glass sponges are ecosystem engineers. Their grazing rates, their water processing rates, that is the rate at which they do what they do, are up to 10 times higher than other suspension feeding communities. Now, what do I mean by water processing rates? Last week, Amanda showed a video, I'll just show a still. What you don't see in this photo is that a diver has just injected some green dye at the base of this rather large barrel sponge. And it's amazing just how quickly and how strongly this green dye that came in at the base is ejected at the top. That's how fast this sponge is processing water. And we can tell because the dye is in the water. By the way, non-toxic dye. So whatever it is these sponges are doing, they're doing it a whole lot. There are also carbon sinks and ammonia sources. And I don't want to gloss over that because both of these phrases are of great ecological importance, especially these days. And I want to make sure that, that we understand just what they mean. Carbon sink, I'm sure we all know what photosynthesis is. That's where green plants do what they do and take what they need out of the atmosphere. And what they need is carbon dioxide. So when we say a carbon sink, that is something that takes carbon away and hoards it, we mean ultimately carbon dioxide that came from the atmosphere. When it enters the ocean environment, it gets photosynthesized and it turns into biomass. It turns into more photosynthesizer, which possibly gets eaten, by bigger and bigger things, and things move around the food web. But eventually this carbon might get sequestered. It might get grabbed by the sponge. And so we love carbon sinks because anything that's a carbon sink is taking carbon dioxide out of the atmosphere. And that carbon dioxide, our name for it these days, is greenhouse gas. So anything that is a carbon sink definitely should be encouraged, studied, protected. Maybe you already knew that, but I think less understood is the benefit of something being an ammonia source. Ammonia is the starting building block for the kind of molecule that people in my business, bioinformatics, care about very much. We care most about DNA and RNA and proteins, and all of these are long backbone molecules, and the backbone needs nitrogen. And the nitrogen comes from ammonia. And the ammonia ultimately comes from the atmosphere. Most of the atmosphere is nitrogen triple bonded to nitrogen. Here's my diagram of triple bonded nitrogen. And breaking that triple bond is the most energetically expensive reaction in all of nature. It's difficult to do. The only other common triple bond that we know about is acetylene. And if you think about the last time you saw an acetylene torch, maybe as you walk by the science building being built, the kind of energy that it takes to break those bonds and the kind of energy that's released. Well, similar to acetylene torch, but imagine doing that if you're a microbe. Very few organisms break atmospheric nitrogen and create ammonia that can turn into life. So it sounds like this is just one more vital process, but it's a bit more than that. It's more like the accelerator. It's the throttle because what we found is in the ocean, when there is creation of ammonia from atmospheric nitrogen, then almost always there's abundant life. Ammonia, fixed nitrogen, is the limiting nutrient in the ocean. And so, you know, when I learned that sponges are great ammonia sources, that is to say, they're creating abundant life, that's good in its own right. And also much of this abundant life is photosynthesizers, which are carbon sinks. See previous discussion about we like carbon sinks. Another thing about glass sponges is they form huge reefs. This map, well, the inset will show you where it is. It's in the Georgia Sound between Vancouver Island and Vancouver City in the mainland of British Columbia. And all the red dots are huge reefs formed by glass corals. And this is why in my first bullet, I say that, I'm sorry, glass sponges. I say that glass sponges, like corals, are ecosystem engineers. The skeletons of these corals are biosynthetic glass spicules. To me, it's amazing that that any life can synthesize glass, but there's enough silicon dissolved in the ocean water that that they can do this. These are their skeletons. Now, imagine your skeleton or my skeleton after we are laid to rest. Who knows how long that skeleton will last? But glass will last a lot longer. Now, when sea temperatures rise, protect the corals, but bet on the sponges. They have a better chance. How do we know this? Well, one way we know this is the last time we had sea surface temperature rises like we've got going on these days was 3 million years ago during the early Pliocene warm period. Core samples from the Bering Sea tell us about how sponge population changes during those times. And the short answer is that sponges are successful. Now, think about what would be in a core sample that's 3 million years old. Nothing from you and nothing from me could possibly survive, except maybe, see, I'm kind of wearing this glass dolphin medallion. Maybe little particles of my glass will survive, but nothing of me. Glass can last a long time, especially in microscopic form. So it's really important that we study many of these core samples. Each of these samples contain spicules and many sediment grades. By the way, if you look at the credit at the bottom of this map, Aiello and Ravello, this is Amanda and my colleague, Dr. Aiello from Moss Landing Marine Lab. Now, what we get out of these core samples is too much information for manual analysis. The prevailing piece of scientific apparatus for analyzing slices of a core sample is the graduate student. But if you imagine that even the very smallest core samples are an inch in diameter, and this picture I'm showing here with lots of interesting stuff going on is less than a millimeter by less than a millimeter, you can see how there aren't enough graduate students in the world to scale up this analysis to the point where we can really get deep knowledge out. So the plan is to use flow imaging microscopy and couple that to deep learning. Now, a flow cell, in its simplest form, is simply something where all the different particles in the sediment drip one by one past some optics, and a photograph gets taken of them one by one. And in the case of our study, what gets photographed is particles from the kind of thing whose particles survive for a couple million years, sponges and diatoms and radiolarians, which are other silicon-using critters of the deep. Now, the challenge, what we want to do is classify each image of an individual particle as either sponge spicule or not a sponge spicule. For extra credit, taxonomic identification of the spicules. Now, the speed bump is that flow cam that actually takes the pictures. We don't have one. What we've got is a few dozen photographs from a courtesy run that the manufacturer did for us, where we've got a dozen or so spicules, maybe a little more. We've got some diatoms, we've got some radiolarians. In other words, on the left, we've got the stuff that we're trying to classify. In the middle and on the right, we've got the decoys. There's a technique in deep learning called data augmentation. The closer you are to a computer scientist, the more this will spin your head around because it seems like it couldn't possibly work. We're using the kind of deep learning that's been optimized for image processing. And to train these deep learning models, we need lots of images. The 173 that we started with were enough to get started, but definitely not enough to finish. But it turns out that you can take your images, you can flip them, as in the top, you can rotate them, as in the middle, and you can even skew them, as in the bottom. Skewing is like printing them on rubber and then stretching out the rubber. And so we did that, and our 173 images became 12,000 something. Now, those of you with a background in information science are looking at this and saying, you have not added any information. And I respond, yes, I know, it bothers me too. But it turns out that by adding these same information content images at different angles, at different skews, we are giving the model more opportunities to make the right call in ways that I think it would not be appropriate to get into now. So instead of doing that, I'm going to show you the results. 4 ways to evaluate performance of a classifier. We've got test accuracy, F-score, precision, recall. Kind of doesn't matter which one you look at, does it? They're all pretty much the same. So let's look over on the left. And what we did with this is we tried a different approach called support vector machines, just because it's a logical approach. It's remotely related to neural networks. We didn't want to just say, well, we tried neural networks and they did okay. If support vector machines are going to be better, then we wanted to use them. But as we see on the right, the gold and green bars show that these are results with neural networks. On the left, we have support vector machines. So thank you for coming, support vector machines, but we'll be using neural networks. Now, the gold bar with the red star is our results with the original small data set. Slightly over, let's call it 91% accuracy. All right, that's not bad. That would be enough to say, let's keep going and try to improve it. But with the enhanced data set, with the transformed data set, we're just around 95% accuracy. That's at the point where we're saying, you know what, if we had a bigger training set, actually genuinely more different photographs of stuff, we're quite confident that this would work. So we published this. And remember I said, I talked about the challenge and so we're able to classify images as sediment, spicule, other stuff. We're now working on the extra credit, taxonomic identification of spicules. This seems natural enough once we can say this is a spicule. It would be great to be able to say, and what kind of sponge did it come from? Also, if we can do this, we can take FlowCam on board ship and analyze water in real time as we sail through it. And this would be a huge advantage. Peripheral vision, check. Now let's talk about coral vision. This is by far our harder problem. And as you'll be able to see from the sophistication of my slides, we've solved not quite half of it, but we're very happy with the solution and the approach that we've got. Let's motivate this by talking about corals. Corals fringe about 1/6 of the world's coastlines, and they support millions of species, including one species I very much care about, humans. Well over 1 billion people get most of their protein from the ocean. And much of that protein requires coral reefs at some point in its development. If we lose the coral reefs, well, hopefully sponges will take up some of the slack, but we ought also to protect the coral reefs. Coral reefs are places of stunning beauty, and I feel so lucky that my career involves studying these. Reefs have direct economic importance, mostly from tourism, that would be scuba and snorkeling, and from fishing. The estimated direct income is 48 billion annually, and if that sounds like a nice income, all you have to do is take $1.2 trillion, invest it at prime rate, and you'll make your $48 billion annually. Although it's with mixed feelings that I even showed this slide, because if you put a price tag on something, someone will come and pay it. And the actual value of coral reefs is far beyond anything monetary because of their ecological importance. If we lose all the coral reefs, our chances of survival on this planet are quite dim. Coral reefs are places of concentrated biodiversity. It's no exaggeration to say that these are rainforests of the ocean. They're hotspots of nitrogen fixation, that is creating ammonia out of atmospheric nitrogen. They're hotspots of photosynthesis. So we've already talked about why that's ecologically necessary and desirable. They also shelter juvenile fish. Imagine if you're a tiny fish trying to get away from all those predators. There is nowhere better to hide than the tiny nooks and crannies and convolutions of a coral reef. And the threat to reefs are many. Pollution and destructive fishing are two. Climate change is the one that I study. There are two components to climate change threats, and one is ocean warming. What ocean warming does is it drives out the symbiotic microbes that live within the coral polyps. So we see this purple image on the left and we think, oh, look at those beautiful purple coral polyps. Actually, no. What's purple is the microbes that live in association with the polyps, and they can't handle too much heat. When the water temperature gets too high, the microbes move out, the corals get bleached, and the polyps starve. Now, corals can recover from events like this, but not from too much of it. The other threat is ocean acidification. Greenhouse carbon dioxide enters the ocean by a process that's similar to how we make soda. If you've never made soda, all you do is you take some water or some soda syrup, and you increase the carbon dioxide pressure in the air above it. This drives carbon dioxide down the CO2 gradient into the water, where CO2 combines with water to form carbonic acid. This is why the name for fizzy drinks is carbonated drinks. Carbonic acid, focus on the word acid. This increases ocean acidity. Carbonic acid is a weak acid, but even so, it's strong enough to degrade calcium-based shells. So it's the same process that carbonates soda. Unfortunately, this planet is putting the fizz into ocean physics, and we need to take it out again. Carbon-based shells are degraded. That's the threat to free-living shellfish, to coral polyp shells, and what gets made out of coral polyp shells, reef substructures. And what we see in this picture is coral reef rubble from a reef that has collapsed, and it's possible that it collapsed because of erosion of etching away of the structures of the coral polyp shells. What do we do about this? Well, we can't protect, we can't study what we can't measure. This is a device called an ARMS, Autonomous Reef Monitoring Structure. Of course, it's autonomous. All it does is sit there. These are wonderful because they're cheap to make, relatively easy to deploy. You just take it, you bolt it to the ocean floor near a reef whose biodiversity you want to measure. Or if you want to measure something else, you don't have to put it near a reef. There's the reef. And actually, good practice is to replicate your experiment. When it goes down, it's a pile of plastic. When it comes up, it's a habitat. Now, analysis can be visual or genetic. Visual means you get an expert who looks at everything and dictates to somebody with a clipboard and says, you got 3 of those, you got 5 of those. Genetic is where you extract DNA, and through various bioinformatics approaches, you analyze that DNA. This is low cost at low scale. All you really need is cheap genetic analysis equipment or access to a really good taxonomist with local expertise. It's impossible at high scale. And also, it doesn't do a good job at measuring the mass of the individuals that recruit to the plates. You can see on the bottom right a plate with some individuals recruited to it. So it doesn't scale up. This is too bad for the RAMP, the Reef Assessment and Monitoring Program that NOAA executed in the Pacific from 2006 to 2018. They deployed hundreds of units at 15 meters throughout the Pacific Ocean. Hundreds of units means thousands of plates. And obviously, identification by a human taxonomist or genetic ID, they just don't scale up this big. Fortunately, they took photographs of every single plate. And so deep learning plus ARMS photos is the Coral Vision project. The big idea is to develop and deploy neural networks for analyzing these photos and enable the whole global research community to use and improve our neural network, to access large databases, and to contribute to databases. When you have images and data and exploration, you have engines for generating hypothesis-based research. By the way, what you also have is a way for citizen scientists to get, without hypocrisy, a genuine sense of contributing to the solution. I believe that there's tremendous energy among people of all ages, and perhaps especially concentrated in our K through 12 schools, that are eager to do something about the world they're inheriting. They just don't know what to do. What we have is our 14,000 plates, the photos, and the metadata. That would be, where, when, how deep, and so on. Easy enough to get external metadata, sea surface temperature, acidity. And fortunately, because I work at San Jose State, I have a motivated, talented core of graduate and undergraduate research students. Step one is called segmentation. This is analysis of a photo to find the individuals. On the left, we have original photo. On the right, we have false image of 3 individuals that have been found by our software. You can see that it did a spectacular job on the blue thing. On the right, it's blue. Not so well, not horrible, but not great on the others. But this is a very good beginning, and what we now need to do is identify on all our 14,000 images which individuals have been well segmented. The way we want to do this is have volunteers. I'm working on a software system that will let them just inspect side by side as you see the original and analyzed images, and the volunteers will click on cleanly segmented individuals, which will be collected programmatically. And step 2 is to take these individuals, they are now extracted photos of individuals, and cluster them by visible traits, by phenotype. And hopefully what we'll get will be each one categorized by their species. That's the hope. Probably we can't do better than genus. I'll certainly accept it if we can do family. And step 3 is to show the clusters to expert taxonomists. They'll assign taxonomic classification, and notice how low the burden is. A few kinds of individuals, the best representatives, from each of a few dozen clusters is possible, versus the impossibly high burden of having taxonomists identify every individual in each of 14,000 plates. Next step is to build a deep learning classifier for all the individuals that we find in the plates. And this will look roughly like the handwriting classifier, except that instead of digits, we'll have extracted photos of individuals. And when a number on the right lights up, turns on, it won't mean I saw digit 9, it means I saw Redis blobus. Step 5 is apply the classifier to the images. This is straightforward. All it's going to do is tie down a bunch of nodes on the HPC that we own for several months. Step 6 is what I think of as the magic, the reason for doing it. Citizen science. We want to create a geographic website, kind of like the Johns Hopkins COVID-1 maps. And any user, just by clicking on a part of the world, can explore our data, analyze our data, form and test hypotheses. Any user can do that. Qualified users, for example, teachers, and I'm already working with some in San Diego at a high school, can upload their own data for analysis by Coral Vision. And then super qualified users like the Smithsonian Environmental Research Center, who they do a lot of arms experiments. They may contribute to the database. There's also been interest from KAUST, that's the King Abdullah University of Saudi Technology. They're tasked with protecting the biodiversity of the Red Sea as Saudi Arabia moves from an oil-based economy to a green tourism-based economy. Well, here are the gratitude slides. These are my partners in crime for peripheral vision and my partners in crime for Coral Vision. And if you look at all of them, you can see that they look like really nice people who are really happy doing what they do. And a lot of that has rubbed off on me. I'm so happy that I get to do what I do. I'm so happy I got to share it with you. And now I will be very happy to take any questions.
Thank you, Phil. A virtual round of applause for your talk. It was really great. There are no, let's see, there was a question early on that I'm afraid I didn't get to. It was a question about whether the bias of one layer becomes the input for the next layer. And I believe that's the standard way that, is that not right? Okay, why don't you answer? It has to do with the structure of the machine learning.
Yeah, so the sum of the weights of all the active inputs is compared to the bias. And in the simplest kind of neural network, the output fires or doesn't fire, depending on if the sum of the weights is greater than or less than the bias. But in this simplest example, the output is always 0 or 1. That means the next layer's input is always 0 or 1.
Got it. I'm going to take host prerogative here and ask a question before I pass it on to the next people. So there's a project in the astronomy world where astronomers posted huge numbers of galaxy images and then asked amateurs to essentially classify them into a group of different sort of galaxy types. Have you thought about doing a more like brute force method like this, where you have individual non-experts try to classify things and see how that compares with what the machine can do?
That would be a really good, well, let me think about that. If non-experts are doing classification, I wouldn't want to weigh a non-expert's classification versus a machine's classification. I would want to involve a human in that. It could even turn into a kind of a contest. Can you classify better than a machine? Can you classify better than an expert? But I've been very sensitive to the necessity to reduce the burden on human taxonomists. So what I want to do is have just civilian participants saying, yeah, you partitioned this one right, you partitioned this one wrong, but for the identification, I want to get the best training I possibly can.
Got it. Okay, so what I'm going to do is, as I see questions in the Q&A, I'm going to open up that person's audio so they can ask the question themselves. So the first questioner is Amanda Kahn.
Hey, Amanda.
Hi, Phil.
Hi.
Great talk.
Thank you.
That was really exciting. I'm familiar with the peripheral vision, so my question is about Coral Vision. How dependent is Coral Vision on the scale or zoom of an image and on the quality? And just as follow-up to that, is there a way that you normalize or standardize for that?
Yeah, we normalize the images. We do contrast correction, sort of spectrum correction to keep the colors within a certain range, the brightness and the darkness within a certain range. As for the size, as long as the neural network gets images at many different scales, it can classify correctly. An alternative is to try to scale all the images of Redis globus to be about the same size as one another so that we don't present that challenge to the network. But we're not there yet. And what I'd like to do is see if the network can just handle various sizes, because of course these organisms are all different kinds of sizes. See if the network can handle that, because I honestly don't think it would be necessary to scale, especially because size doesn't seem to be an important phenotype for classifying.
Great, thanks very much.
Can I follow up on that and say, is shape an important phenotype for these organisms?
For some, yes, and for some it isn't. They're just blobs, and
Right.
and the most important phenotype is some combination of color and texture. Okay. Texture is really hard to model. A lot of work with neural networks is about stuff that got photographed on dry land. And for that, you can do this thing called transfer learning, where you take a mostly trained neural network, something that's good at noticing stuff in photos, and you complete its training so that it becomes a specialist. But what we found is that these starter kits do great on dry land, but they're not so good for stuff whose shape is dictated by life in the water.
Thanks. Okay, thanks, Amanda. Our next question is from Bill Andriopoulos. So let me open up Bill's talking. There you go. Bill, you have to unmute yourself. There you go.
Hi, Phil.
Hey.
Thanks for the excellent talk. I was just wondering, why is identification by genetic information impossible in the sense that it doesn't scale?
So classical, by genetic?
Yeah, I thought I think you said that by the DNA sequence, identification of the species wouldn't scale.
Yeah, it would require having somebody process every plate manually and extracting a tissue sample from every individual. That's the first problem. And actually, there are people who do that on a small scale. And that's one thing I was doing in Moss Landing. But, and forgive me, everyone, for using technical language that I know Bill and I share in common. It turns out that there's enough haplotype variation that a big common database like GenBank isn't good enough for identification. You need a really good reference database that comes from the part of the world that you're sampling. And those reference databases are really expensive to build. And once you've got one, you can then one by one analyze the organisms on your plate until you've got too many plates to analyze.
And is there enough genetic variation from location to location that set only works, say, in a certain reef or?
Nobody knows. We suspect there are reefs that are only sufficiently like maybe themselves or maybe a few other reefs. What you don't want to do is say, oh, this is this particular species. It's not especially like it, but it's my closest match. Well, it's your closest match to Monterey Bay, but its closest match to something in Hawaii would be totally different, but it's not in your database.
So with the deep learning methodology, could you differentiate between strains, like something from Monterey Bay versus Hawaii?
Oh, Bill, I'd love to. I really doubt it. I don't think the resolution of this is the strain level or the genus level. I'd be very happy if it can resolve to genus level, and I'll settle for family level. A lot of strain differences are cryptic species, which means that human taxonomists can't tell the difference by looking at them, which means that no model based on visible phenotypes can do any better than that.
Yeah, that's what I thought. Okay.
Yeah.
Thank you very much.
Okay. Let's see. Wendy Lee has just posted a question. Let me open up her audio. There you go, Wendy.
Hey, Wendy.
She has to unmute. There we go.
It's a great talk. Thank you so much. I invited my whole family here. I want to make sure that the boys are quiet when I ask this question.
Hi, Don. Hi, kids.
It's really impressive, and I can see how daunting this job is. One of the questions that I have kind of follow on Bill's question is that since we analyze genetic material so much that, you know, how correlated or how much can you look at the phenotypes and the appearance? Can you say, you know, pretty accurately that this is the genetics that this organism have, or is there any other really closely, you know, genetically closely related organism also share really similar phenotypes but they're still distinct?
Yeah. So all I know is I was involved in one project at Moss Landing, and we didn't have the right reference database of nucleotide sequences. We were doing CO1 barcoding, and we just didn't have the right reference database, and we weren't getting clear results, which is all I know, because even if DNA barcoding could just be perfected, the cost would be prohibitive, and it still wouldn't scale up, because you can only sequence what you can sample. And if there are several hundred or even several thousand individuals on a single plate, that won't scale up. There are people who do a meta barcoding scrape, where you just sort of take a paint scraper, scrape everything off the plate into a blender, and you make DNA slurry, and you sequence all of that. This has been done, but when you do that, you lose a lot of information. You lose information about what DNA molecule was on what part of the plate next to what other individual. And you can't do biodiversity statistics, because you can no longer count biomass of every species or the number of every species.
Thank you.
Thank you, Bioinformatics faculty core. Michael, you're muted.
I failed the Zoom test. So there are no more questions in the Q&A or in the chat. So what I'd like to do is take this opportunity to thank Phil for giving us a great talk. And actually, maybe Phil, for the final 30 seconds, maybe you could tell us about this critter that's on the screen.
This is a cuttlefish. My wife and I were diving on the Great Barrier Reef in January of last year. And one thing I can tell you about him or her is that it was a commercial trip. So there was a photographer taking a picture, and there's a bigger shot of me with the cuttlefish. The photographer took the shot, signaled me to move out of the frame. My wife moved in, and by the time she had moved in for her shot, the cuttlefish had changed color. And it was bigger than a large pizza, more like an extra large pizza.
Great.
Thank you so much for this opportunity. Thank you, everybody, for coming.
All right, thank you, Phil. And I'm sure Phil would be happy to answer additional questions if you email him after the fact. Thanks, Phil. We'll be in touch. Bye, everyone. Thanks for joining us.